Well, thanks for that introduction, Chris. So, as Chris mentioned, I'd like to talk to you today about some of the work that we do in the Antarctic Geoscience Group. In particular, looking at the variations in the benthic habitats and the diversity of the seafloor communities that live there. So first of all, I'd just like to give you a little bit of context as to why we are interested in Antarctica. It's not just the fact that there are some really cool things to see, like penguins and icebergs, but we actually have some good reasons to be there. Also, I'd just like to give you a bit of an introduction as to what's so special about Antarctic marine life, what makes it unique in the world's oceans. And then I'll go through a couple of case studies that we've done over recent years within our program to understand marine life in Antarctica and to talk to you about how this work contributes to conservation efforts along the Antarctic margin. And then at the end I'll just briefly touch on some of our future priorities. So first of all, why do we care about Antarctica? Well, one of the most important reasons is that we actually claim 42% of the Antarctic continent as our territory. This is equivalent to 77% of the size of the Australian landmass, so it's a huge area that we claim ownership of. And as well as that land area, we have the adjacent marine jurisdiction, which adds 2.2 million kilometres to our marine jurisdiction. So this is an area that extends from 45 degrees east over here, right across to 160 degrees east, with a little part of it cut out where French has its territory. So this gives us a huge responsibility for looking after this environment. In 2009, the Australian government came out with a number of key policy priorities which really underpins the work that we do in Antarctica. The first of these was to maintain and increase our physical presence, because this demonstrates our effective sovereignty over our territory. The second was to maintain a diplomatic presence, and we do this through engagement with the Antarctic Treaty System and other international instruments. The third was that they wanted us to improve environmental management in the Australian Antarctic Territory and Southern Ocean, and to deliver scientific outputs that meet the defined policy needs of government. The fifth one there is to pursue international collaborative science with states active in Antarctica, and lastly to pursue appropriate economic opportunities, so this includes things such as tourism and sustainable fisheries. So the two areas that our work really contributes to is improving environmental management and delivering scientific outputs for policy needs. So I'm sure when a lot of you think about Antarctic life, you think about the cute and cuddly things like penguins, smelly things like elephant seals, maybe whales, especially with the recent rulings on whaling in Antarctic waters. And these are all lovely, but what I'd really like to talk to you about today is what lives at the bottom of the ocean. You might not have thought much about what lives at the bottom of the sea, but certainly before I first started in this project, I thought, it's just mud really, isn't it? There's not much there. But hopefully you'll come away today with a new appreciation for what's living at the seafloor. And also a new appreciation for just how tough it is for these organisms to survive on the Antarctic seafloor. There's a number of conditions that make it really tough for these organisms. And the first of these is that for much of the year there's really low light levels on the Antarctic seafloor. This image up the top shows the winter sea ice extent, which covers a massive part of the ocean during the winter and is quite effective at blocking sunlight to those surface waters. And as you can see in this figure here, we go from a minimum in sea ice extent during February, March of just under 5 million kilometres squared. This increases through the autumn and winter time and the early part of spring to a peak in sea ice extent in September and October of close to 20 million square kilometres. So we've had a fourfold increase in sea ice extent over that annual cycle. So what this means for those organisms that live at the surface of the ocean and rely on photosynthesis is that we get a significant decline in productivity during a large part of the year when we have this high sea ice concentration. Coupled with that, we have the seasonal cycle of sunlight, which, as you know, means that there's less sunlight in the wintertime, and in fact the sun doesn't even rise as we get below the Arctic Circle. And so if you take these two factors together, we've got very low light levels for about 6 months of the year. So this means very low production in those surface waters, and so those organisms that are living on the seafloor waiting for that food to come from the surface waters are facing about 6 months of starvation each year. Another tough condition that these organisms have to deal with is the extreme cold. It's so cold that you get these chimneys of ice called brinicles that come down from the surface ocean. These are formed from super saline, super cooled water, come down from the surface and spread out along the seafloor, encasing those organisms in ice. So it gives a whole new meaning to a cold snap. But the cold also has some advantages. It means that the typical range of marine predators that you see elsewhere in the world's oceans can't survive in the Antarctic. So this includes organisms such as sharks, crabs and lobsters. So instead what we have as the main predators on the Antarctic seafloor are things like sea stars, sea spiders and ribbon worms. Now the important thing to note about these organisms is that they're fairly slow moving, and they also have no crushing ability. So most of the organisms that live on the seafloor around the Antarctic are soft-bodied. They don't have the hard shells that you see elsewhere to protect themselves, and we get quite a different structure to these ecosystems. Now, a final factor that I'd like to tell you about is iceberg scouring. Icebergs act as bulldozers, and they essentially scrape the seafloor clean in water depths of up to 500 metres. This is an image that was taken at just over 500 metres, 550 metres on the seafloor, and you can see that there's nothing left. It's all just been wiped clean. But what this means is that some of those faster moving organisms, there's some brittle stars here, get the opportunity to recolonise these new areas of seafloor. So it can actually act to promote diversity by opening up new areas of seafloor and allowing new species to move into an area where they might not have been previously. So it's not all bad news for the organisms, but you might think that, you know, with this whole combination of really tough conditions, that there really might not be much that can survive in these waters. But what we find when we go down there is an absolute bonanza of sea life, and this is just completely unexpected. That we actually have very high diversity in the Antarctic, and in some areas it actually rivals that we see in tropical reefs. So this is a seafloor photo that was taken in an area just south of Tasmania, near the Mertz Glacier. And what we have here is a whole collection of these deep cold water corals, some hydrocorals shown in the pinks and the whites here, and a whole diversity of other organisms associated with this ecosystem. Range of sponges and bryozoans and soft corals, this really cute little octopus that was swimming through. And these are also forming substrate for a number of other organisms, such as these brittle stars, which are just smothering these other animals, and little feather stars or crinoids, which are perching up on top. So it's really astounding the diversity that we actually see. Just a couple of other examples. This is from a near coastal area. And we have this beautiful sponge and soft coral garden. And a final example, we have a high diversity of sponges and bryozoans here, and also some hydroids. So we get a range of different ecosystems on the seafloor. So we know that there's a lot of diversity in the Antarctic, but what we really need to do to be able to contribute to conservation is to know how these ecosystems are distributed at a broader scale. And it's essentially going to be impossible to do this by, you know, towing cameras over the entire Antarctic margin. So we need some smarter tools to be able to understand how these ecosystems are distributed. And so that's how the work that we do at GA can really help to fill out this picture. What we do when we go on survey is use a variety of tools to map the seafloor environments. The primary tool that we use is multi-beam sonar, which gives us a really high resolution map of that seafloor bathymetry. which allows us to get an understanding of the different environments and the processes that have shaped the seafloor. So as an example, you can map out these shallow bank areas and then down into some deep basins and look at the features in between that have shaped them. In addition, we can collect sediment samples to understand the composition of the seafloor sediments and the grain size properties. And then we can integrate this physical information with the biological data. So often we'll undertake towed video surveys to get an understanding of the distribution of the different ecosystems. Occasionally we'll also integrate with dive programs that have taken place in shallower water areas. So where have we undertaken these types of studies? Well, I'll take you through a couple of examples that we've done in recent years, starting off the George V shelf, which we had a survey in 2008, then coming up to the coastal region off Australia's Davis Station, then coming into the Amory Ice Shelf just a little bit further west, and then I'll just briefly talk to you about some recent work that we were involved in earlier this year off the Totten Glacier. So the George V shelf is that area just directly south of Tasmania, and we were lucky enough to be involved in a research program here as part of the Census of Antarctic Marine Life back in 2008. And we deployed GA's deep underwater camera, DUC-2, to get an understanding of the seafloor environments and the ecosystems in this area. We were lucky that we had quite a detailed bathymetry map that we could use to target our sampling. And so we were able to sample across a broad range of environments from these shallow water areas, this one's at 177 metres, right down to this deepest site, 1175 metres, just adjacent to the Mertz Glacier tongue. And just to show you what these places look like, this is just a snapshot taken from that underwater video at 177 metres, and this one at 1175 metres. So we get two completely different biological communities at these water depths, but also we can look at the range of different environments across this region. We have a range of coastal sites here which are in these really ruggedly eroded coastal basins. Then we come into this deep George V basin, and then we have these shallow bank areas which are around the 200 to 300 metre mark, and then out onto the edge of the continental shelf we had some sites, and particularly ones that are associated with some of these canyons here which were important to investigate. In addition, we took a whole range of physical samples, so this allowed us to get an understanding of the sediment properties. So this map here shows the mud content across the area. What we see is that this helps to understand more these environments that we have. So the deep basins, such as the George V Basin and the Adelie Basin over here, they form depot centres for the deposition of these muddy sediments, so we have high mud content in these basins. As we come up onto the banks, such as the Adelie Bank here, we have much coarser grain size because those fine sediments have been winnowed by the currents and the action of icebergs. So we can integrate the physical data that we were able to collect, the bathymetry data, the sediment data, the composition of the sediments, and the biological data that we had from those towed video surveys to investigate what might be driving the distribution of the seafloor communities. What we found was that there were three variables that were highly significant in explaining the distribution of these communities. These were the water depth, the seafloor relief, and the mud content. And these had a correlation to the biota of 0.71, which is actually, might not sound high for some of your studies, but for biological work this is actually quite a strong correlation. The other aspect that we wanted to investigate with this work was the impact of the seabed morphology on shaping these marine communities, and I'll just take you through the broad-scale geomorphic features across this margin. So we start off in the abyssal plain, that deep, fairly flat area of the seafloor. And then we come up into the lower slope area and the upper slope, which is the steeper part of that slope. And these contain a whole complex pattern of canyons. And then we come up onto the continental shelf where we have shelf banks that are intersected by these deep basins. And in some areas, these basins are actually connected to the slope areas by these cross-shelf valleys. So this provides a connection between the productive shelf waters and these slope environments. The other thing to just note here is that we have some canyons that come right up to the top of the continental shelf rather. And these are important for environments for the ecosystems. They've been shown to have high current flow, which provides upwelling of nutrients, and also the availability of hard substrates within the current canyon walls provides places for those organisms to attach to. So I'm just going to show you a couple of snapshots of what we find in a couple of these geomorphic features. Starting off with this deep basin here, we have a site here at 800 metres, and as I said, we have really muddy sediments in this deep basin. You can see that in this image here. We have these thick mud deposits that have been burrowed into by a range of infauna, and you also get some of the mobile deposit feeders which crawl about in these environments. As we come out to the edge of the continental shelf here, or the intersection of this cross-shelf valley and this shelf-cutting canyon, we get that community that I showed you before, this hydrocoral, rich, diverse community at a very similar water depth, 725 metres water depth compared to 800 metres in the deep basin. These organisms are ones that attach to the seafloor, and they rely on the food that's being swept past them. So it's telling us that we have a lot of food coming out through this shelf valley and most likely up through this canyon system, supporting this very rich filter feeder community. As we move further along this continental shelf, away from the cross-shelf valley and away from the shelf-cutting canyons, we get quite a sparse community, again at a very similar water depth. There's a sparse, I'm not sure how well you can see it, but there are just sparse occurrences of these little ball sponges and a couple of fish. So it's much lower diversity than what we see in association with these canyons and cross-shelf valleys. So this is indicating that the geomorphic features are a good way of understanding how these communities are distributed. So I'm now going to take you across to Davis, where we had a survey a couple of years ago that Phil O'Brien led, and this was the first time that this seabed environment was ever mapped using multi-beam bathymetry. So it revealed a range of environments. We have these rocky outcrops on the seafloor, have some really heavily iceberg-scoured regions, some deep basins, and Phil was able to run a number of video tows across this area to look at the seafloor communities. Since then, Jodie Smith has been working up this data, and she's found that there's a strong correlation between the biota and two main physical properties. These are depth and the backscatter that we get back from the multi-beam data. And the backscatter is really telling us about the substrate type. So whether it's muddy, whether it's hard, or whether it's somewhere in between. And so again, we're getting sediment properties and depth together, giving us a strong correlation to how these benthic communities are distributed. The next case study that I'd like to take you to is something completely different. This is the Amory ice shelf. And just in case you're not quite sure, an ice shelf is an area of permanently floating ice over the seafloor. And the Amory ice shelf is about 300 to 400 metres thick, and then you have several hundred metres of water column before you get to the seafloor. Now the aim of this work was to try and drill through those hundreds of metres of ice to try and look at what's happening beneath the ice shelf and on the seafloor. This took many field seasons. You can see that this spans 2003 to 2010 to drill 6 sites on this ice shelf. And it involves using a hot water drill. So this is a tent here that they're setting up to do the hot water drilling. Hot water drilling obviously involves a lot of water, so they had to collect ice in these holding tanks, which they then melted down to circulate around that drill so that they could use it to make a hole through this ice. So this is a picture through the ice hole. It's about 20 centimetres in diameter, and they had to try and keep this hole open for long enough to lower various instruments down through the hole to measure the properties in the water columns. They were looking at the oceanographic parameters, as well as to lower down a purposely built sediment corer, which Craig Wintle here at GA designed. And they also lowered a seafloor camera to have a look at what might be living there. Previous studies of ice shelves had indicated that there's really not a lot that can survive beneath an ice shelf because they're cut off from any source of primary production. There's no photosynthesis beneath 300 metres of ice, so any of that food source that comes into the ice shelf has to come from the open water. So we put a camera through the ice, but we really weren't expecting to see much from this environment. So the first site where we were able to lower the seafloor camera was 100 kilometres from the edge of the ice sheet, so 100 kilometres from any open water. And surprisingly, they found that there was actually quite a diversity of life there. This picture down the bottom shows a bryozoa living on the seafloor, one of those filter feeders that has to feed out of the water column. At the top here we have a sponge and a little pencil urchin. And they actually found 22 different species surviving beneath this ice shelf, 100 kilometres from any food source. So then, further down in this ice shelf, we come 160 kilometres from open water, and still we're finding life. So here we have a little krill that's been feeding off the seafloor, a little polychaete that was shown in the videos to be travelling across the seafloor in search of food. Have this very strange jellyfish, which I think if I saw it out my window, I'd think it was a UFO. A little polychaete sticking up out of the sediment, and all these little sediment mounds, which are most likely feeding mounds. So obviously we still have a source of food coming into this ice shelf 160 kilometres from the open water. So then, okay, look at 200 kilometres from open water, and still we have this little hard urchin here, have another polychaete, we have more of these feeding mounds, and we have what we think is a little cup sponge. So we're still getting things that are living in this environment. So we turned to the sediment cores to try and get a better understanding of the food source that might be being delivered to these sites. And we looked at the diatom content. The diatom are those surface water algae that are really the prime source of food for the Antarctic benthos. What we've found, these circles here indicate the relative proportion of diatoms in those sediment cores at the surface of those sediment cores. And we found that site that's 100 kilometres from open water, where we had relatively high diversity, actually has quite a lot of diatoms in it. The number of diatoms in this sediment sample is actually fairly similar to what we see in open water parts of Pritz Bay. And in fact, that seafloor community with the 22 species was also similar to what is seen in open waters of Pritz Bay. As we come further into the ice shelf, you can see that the number of diatoms drops off significantly, but it's still sufficient to support those organisms that are living there. And we tied this into the oceanographic modelling that had been done for this ice shelf, and found that it actually fitted quite well with the seasonal patterns of bottom current flow that was bringing in diatoms from the open water regions and through into this ice shelf. So this is quite an extreme example of life existing in really tough conditions, but it shows that you can get biota in these conditions if there's sufficient current strength to bring in the food for these organisms. So lastly, I'll just very briefly show you some of the work that we did this last season in the region of the Totten Glacier. And this again was an area that had never been investigated previously. So we were able to map the seafloor using multi-beam bathymetry for the first time. And over the next few months, I'll be looking at how we can integrate the physical data sets that we collected, the multi-beam bathymetry, oceanographic information, sediment properties, with the seafloor camera tows that we did, and here's just a snapshot of some of my favourite pictures that we were able to get from the seafloor in this region. So that's very much a work in progress, so watch this space. But what we've seen so far from these detailed case studies is that there are a number of physical parameters that seem to shape the distribution of biota on the Antarctic seafloor. The primary ones are the water depth, the substrate type in terms of the composition and the grain size, the seafloor relief, the geomorphic features, and the food supply, which can also be approximated by the current flow. But if we want to be able to contribute to conservation efforts, we really need to be able to understand how these ecosystems are distributed at a broader scale. So what we've done is to focus on the physical parameters that we have that are available at a broad scale. And of the significant ones we have depth data available at a broad scale, as well as grain size data, information about the seafloor relief, and the geomorphic features. So we can use these data sets to gain that broad scale understanding of how the organisms might be distributed around the Antarctic margin. So just to take you through what we know about the environments at a broad scale, this is some detailed bathymetry that was compiled by Olivia Wilson from single beam data primarily and a little bit of multi-beam data. We only have less than 1% of the Australian Antarctic territory that's been mapped using multi-beam sonar, but we do have quite a high density of single beam data in a few key areas, such as the areas shown here where we've been able to produce these high resolution bathymetry grids. So outside of these areas where we have the density of single beam data, we have to rely on the satellite bathymetry data. This gives us a fairly good understanding of the bathymetry at these broad scales. We do lose some of the resolution of the detailed features, and I'll just zoom into this area here off Mawson to show you this. So at the top here we have the satellite bathymetry, which is at a resolution of 500 metres, and at the bottom here we have the bathymetry compilation, which was gridded at a resolution of 100 metres. And so you can see that we get a whole lot of features that pop out from this higher resolution bathymetry that we really couldn't see from the satellite bathymetry. So we have these shelf basins which are much more clearly defined in the high resolution bathymetry. But of course we're limited in our coverage with this, so we make do with what we have. But by taking the bathymetry data sets that we have, we've been able to produce a map of the geomorphic features around the entire Antarctic margin, and this allows us to get a better understanding of the different seabed habitats. And I won't take you through this in detail, but the colour scheme is the same as what I took you through over on the George V margin. We've also tried to compile as much of the sediment information as we can over the last few years to look at those substrate properties. Now we've come up with about 700 sediment samples from this area, but as you can see there are some large gaps in our understanding, some areas that are just not accessible due to the persistent sea ice cover. But where we do have these density of sediment samples, it gives us another data set that we can use to understand these habitats. So how does this work contribute to conservation efforts in the Antarctic? Well, over the last couple of years, Australia has put together a proposal for marine protected areas together with the European Union and France, and they've proposed 4 protected areas around the Antarctic margin. And this has been based on both the pelagic, so that's the surface water, and the benthic values. And for the benthic values, it's been based primarily on physical data. The primary data sets are the seabed morphology, that geomorphic features map that I showed you, and the satellite bathymetry data. Now, since these regions were proposed, we've done some further work to compile the sediment data and to produce those high-resolution bathymetry grids to try and get a better understanding of the habitats within these marine protected areas and how well they represent the habitats in the broader region. We want a marine protected area network that actually is representative of this region so that it's doing a good job of protecting the ecosystems that are there. So I'm just going to show you some examples of how well these regions represent the ecosystems and the habitats in the broader East Antarctic region. So first of all, if we look at the sediment properties within the East Antarctic region as a whole, and this shows you the mud content across all those sites that we have samples for in the East Antarctic region. And we'd hope that our marine protected areas also have a similar range of substrate types based on the mud content. And in fact, when we look at just those samples within the marine protected areas, we get a good correspondence with representation across all these different sediment types. If we look at the sand and gravel content, we also get good representation between the East Antarctic in the blue and the marine protected areas in these red bars for both the sand content and the gravel content. Although you'll notice that there's very little gravel in the sediment samples that we have. So how about for the bathymetry? Well, this top graph here shows the water depths across the East Antarctic region as a whole, going from shallow water depths on the right here across to the deep waters, 6,000 metres on the left, and the water depths within the marine protected areas at the bottom here. What we see is that we not only capture a similar range of water depths within the marine protected areas, but also a similar structure in these water depths. We have quite a high concentration of areas that are in this sort of 400 to 600 metre range, which is representative of the East Antarctic area as a whole. And then we get this secondary peak around the 4,000 metre mark, which is also reflected in the East Antarctic region as a whole. The only area that we don't represent well in the marine protected areas are these deeper water areas between 5 and 5,500 metres. So we need to do some more work to find out what this means for capturing the biodiversity of these areas. So lastly, looking at how well the marine protected areas represent the geomorphic features of the region. So this pie chart here shows the proportion of geomorphic features across the whole East Antarctic region, with a large area of abyssal plain and lesser areas of these other key geomorphic features. Now if we overlay what we see protected within the marine protected areas, we get fairly good representation of these main geomorphic features. We get 21% of all abyssal plains are protected within the marine protected areas, 64% of coastal terrains, 59% of ice shelf cavities, 42% of areas of lower slope, 38% of the areas of plateau slopes, 51% of shelf banks, 63% of shelf deeps, and 39% of upper slopes. So we're doing quite well at capturing these different geomorphic features within the marine protected area network. The feature that we don't capture well is these areas of rough seafloor. And these correspond to those deeper areas of 5 to 5,500 metre water depths. And so again, we need to get a better understanding of, well, what are the biodiversity characteristics of these deep areas of rough seafloor. So to summarise, hopefully I've given you an understanding of Antarctic ecosystems and just their diversity and their uniqueness in the world's oceans. In the future, our role at GA is to expand our knowledge of seafloor habitats, and we hope to do this in areas where at the moment we have little information, those large gaps in our data in both the detailed bathymetry and in the sediment composition, in our knowledge of the seabed biota. And that'll allow us to continue to provide good advice to policymakers so that these ecosystems can be protected into the future. If you have any questions, I'd be happy to take them. Thanks.